Parasitic plants use stolen genes to make them better parasites. Some parasitic plants steal genetic material from their host plants and use the stolen genes to more effectively siphon off the host's nutrients. A new study reveals that the parasitic plant daughter has stolen a large amount of genetic material from its hosts, including over 100 functional genes, through a process called horizontal gene transfer. Some parasitic plants steal genetic material from their host plants and use the stolen genes to more effectively siphon off the host's nutrients. A new study led by researchers at Penn State and Virginia Tech reveals that the parasitic plant daughter has stolen a large amount of genetic material from its hosts, including over 100 functional genes. These stolen genes contribute to daughter's ability to latch onto and steal nutrients from the host, and even to send genetic weapons back into the host. The new study appears July 22, 2019, in the journal Nature Plants. Horizontal gene transfer, the movement of genetic material from one organism into the genome of another species, is very common in microbes and is a major way that bacteria can acquire antibiotic resistance, said Claude dePamphilis, professor of biology at Penn State and senior author of the study. We don't see many examples of horizontal gene transfer in complex organisms like plants, and when we do see it, the transferred genetic material isn't generally used. In this study, we present the most dramatic case known of functional horizontal gene transfer ever found in complex organisms. Parasitic plants like daughter cannot live on their own by generating energy through photosynthesis. Instead, they use structures called hostoria to tap into a host plant's supply of water and nutrients. Daughter wraps itself around its host plant, growing into its vascular tissue, and often feeds on multiple plants at one time. It can parasitize many different species, wild plants, as well as those of agricultural and horticultural importance. Parasitic plants live very intimately in connection with their host, extracting nutrients, said dePamphilis. But they also get genetic material in the process, and sometimes they incorporate that material into their genome. Previous studies focused on single transferred genes. Here, we use genome-scale datasets about gene expression to determine whether the large amount of genetic material coming over through horizontal gene transfer is actually being used. The research team identified 108 genes that have been added to daughter's genome by horizontal gene transfer and now seem to be functional in the parasite, contributing to hostoria structure, defense responses, and amino acid metabolism. One stolen gene even produces small segments of RNA known as microRNAs that are sent back into the host plant, acting as weapons that may play a role in silencing. The team used rigorous criteria to determine whether the stolen genetic material was likely to be functional. The genes had to be full-length, they had to contain all the necessary parts of a gene, they had to be transcribed into an RNA sequence that later builds proteins, and they had to be expressed in relevant structures. The team also explored the evolution of these transferred genes as additional support for functionality. We compare a gene's genetic sequence with closely related genes and look for a special signature in how that sequence evolves to tell if it's likely to be functional, said dePamphilis. Certain kinds of mutations in a gene do not affect the protein that the gene codes for and therefore do not impact the gene's function. When we see large amounts of these kinds of mutations, as opposed to mutations that might change or disrupt the gene's function, it is strong evidence that natural selection is acting to keep the proteins intact and useful. 18 of the 108 genes appear in all daughter species, suggesting that these genes were originally stolen by the ancestral form of daughter and are maintained in modern species. This is the first time any study has seen evidence that horizontal gene transfers occurred early in the evolution of a parasitic group, said dePamphilis. In this case, 18 of these genes were present in the common ancestor of all the living daughter species, which may have contributed to successful spread of these parasites. The team also identified 42 regions in the daughter genome that appear to result from horizontal gene transfer but do not have any functional genes. Because such a huge quantity of genetic material has come over through horizontal gene transfer, we suspect that the parasitic plants cannot filter what is coming in, said dePamphilis. But natural selection is helping maintain the useful genes and filter out the less useful segments. The researchers are currently investigating how exactly genetic material is being transferred from host to parasite. They would also like to explore whether this transfer is a one-way street or if the host can obtain genetic material from its parasite. We'd love to know how extensive horizontal gene transfer really is, said dePamphilis. We looked at just one of species of daughter, which is just one of